Welcome to the debate. Imagine discovering that, the anxiety you feel, or maybe a physical tremor in your hand, didn't actually start in your brain.
Right, but rather it started with your breakfast.
Exactly. Or, well, the microscopic organisms digesting that breakfast. Because for over a century, modern medical science essentially viewed the human body as this strict top-down hierarchy.
Yeah, the brain was the undisputed executive commander sitting up there in the skull.
Right, and the gastrointestinal tract was frankly just the plumbing. I mean, it was seen as this passive muscular tube designed strictly for digesting food and expelling waste. But over the last 2 decades, that hierarchy has been completely dismantled.
It really has. We're living through this profound paradigm shift where the gut is now recognized as a highly sophisticated sensory organ.
People are calling it the body's second brain. And as we survey the landscape of medical science in 2026, we're faced with a central, defining question about this, the microbiotic gut-brain axis.
The classic question of causation versus correlation. Which way does the biological arrow actually point?
Exactly. I mean, is the gut microbiome a causal driver of neurodegenerative and psychiatric diseases like Parkinson's and major depressive disorder? Or, and this is the crux of it, is gut dysbiosis merely a correlative marker?
Meaning, is it just a downstream side effect of systemic illness or, pharmaceutical meds and dietary changes?
Precisely. Now, I represent the driver position here. I take the stance that the gut is ground zero. When I look at the data, I see the gut as the causative spark for these pathologies. And that's incredibly exciting because it gives us a modifiable lever to revolutionize how we treat the brain.
And I take the position that while the microbiome is this elegant biological marker, portraying it as the primary causal spark is just a leap too far.
A leap too far?
Yeah. We're relying way too heavily on high-magnitude effects in these artificial animal models that frankly fail to translate to humans. Viewing the gut as a therapeutic panacea risks generating this cycle of cruel optimism for patients. In reality, the dysbiosis we see is largely the result of the disease itself.
Well, let me lay out the driver position, which is fundamentally a bottom-up model of pathogenesis. We have to start with the anatomy.
Okay, let's talk wiring.
Right. Because when I first read the literature on the enteric nervous system, the wiring absolutely blew my mind. We always picture the brain barking orders down to the gut. But the vagus nerve, the main physical cable connecting the two, is actually 80% afferent.
Sending signals up.
Yes, 80% of its fibers are sensory, sending information from the gut directly to the brain.
Which is a beautiful piece of evolutionary engineering, certainly. I mean, the gut needs to tell the brain what we've ingested.
It does, but it also establishes the physical infrastructure for ground-up disease. Let's take Parkinson's disease as our primary example. Have you ever looked closely at Brake's hypothesis?
Of course, the staging theory for Parkinson's.
Right. It posits that the disease doesn't just spontaneously originate in the substantia nigra, the brain's movement center. Instead, an environmental trigger causes specific proteins, alpha-synuclein, to misfold down in the nervous system of the gut.
And then they spread.
Exactly, they're prion-like. Think of them like a line of dominoes. They literally knock into healthy proteins, misfolding them too, and they spread transsynaptically from the stomach straight up the vagus nerve and directly into the base of the brain.
That's the structural theory, yes. But a timeline is harder to prove in living humans.
Well, to prove that timeline, that the gut dominoes fall first, we can look at that massive landmark study from April 2026, the Nature Medicine paper from University College London.
Ah, the GBA1 variant study.
Yes. The UCL team looked at this high-risk genetic marker for Parkinson's, the GBA1 variant. They analyzed the microbiomes of healthy controls, Parkinson's patients, and crucially, healthy carriers of this gene.
Right, the asymptomatic group.
Exactly. The bombshell finding was that the healthy carriers, people with zero clinical motor symptoms, whose hands didn't shake, who were completely functional, they already exhibited a specific Parkinson's pattern of 176 microbial shifts in their gut.
Wow.
It's wild to think about. Their microbiome was ringing the alarm bells long before the brain showed any clinical signs of failure. I mean, that demonstrates a causative spark.
I come at it from a different way.
Oh.
Yeah, the top-down or reverse causation framework provides a much more clinically grounded explanation for what we're actually observing. I don't deny that dysbiosis is real, or that those 176 microbial shifts exist in that UCL cohort.
But
I think we're looking at an ecosystem and confusing adaptation for causation. Let's look at the practical, daily reality of Parkinson's disease. Severe constipation and drastically delayed gastric emptying affect up to 80% of patients very early on.
often years before diagnosis, yes.
Exactly. And when you dramatically slow down the transit of food through the intestines, you fundamentally alter the physical and chemical environment of the gut.
You're referring to the changes in the luminal environment?
Yes, precisely. When transit slows, you change the pH, you change the water content, and you alter what nutrients are actually available for the microbes to feed on.
Okay, I see where you're going.
Right, so that altered environment naturally favors the overgrowth of certain bacteria like bifidobacterium. So that microbial shift you're pointing to as a driver, it's actually an intermediate marker of early autonomic nervous system dysfunction.
So you're saying it's a symptom?
The brain and nervous system are failing to move the gut. The microbiome shifts as a biological echo of that failure. It's not the spark that ignited the fire, it's the smoke. And does this biological echo only happen with physical motor diseases? No. We see the exact same reverse causation illusion in behavior conditions like major depressive disorder.
Wait, you think the depressed microbiome is just a side effect of depressed behavior?
Absolutely. I mean, sad mood reactivity profoundly alters human behavior. When people are deeply depressed, it drives massive dietary shifts toward highly processed comfort foods.
High saturated fats, refined sugars, that sort of thing.
Yeah, and it drastically reduces fiber intake. We know definitively that diet accounts for over 50% of microbiome variation, which far outweighs host genetics. Therefore, the so-called depressed microbiome researchers sequence in the lab is in reality simply a reflection of the depressed diet.
I see why you think that, but let me give you a different perspective. We aren't just looking at loose correlations with broad dietary habits anymore. We can trace this down to precise molecular mechanisms.
Precise mechanisms? Like what?
Consider the 2024 Harvard Medical School study regarding Morganella morgani. This is a specific bacterium frequently enriched in the guts of depressed individuals. And the Harvard researchers discovered it doesn't just passively hang out there. It actively does something highly destructive.
Wait, walk me through that. What is it actively doing?
It literally incorporates an industrial pollutant called diethanolamine, or DEA, to create a faulty neurotoxic fatty molecule.
Oh, wow.
Yeah, this specific aberrant molecule acts as a potent immune stimulant. It directly triggers a massive immune response that drives depressive behaviors. And if you want another clear mechanism, consider leaky gut.
Ah, intestinal barrier integrity.
Exactly. When a patient lacks fiber, they lack short-chain fatty acids, primarily one called butyrate. Now, butyrate is essential for upregulating tight junction proteins, like claudin-5.
Right, the structural proteins.
To visualize this, think of claudin-5 as the mortar between the bricks of a castle wall protecting the brain. Without enough butyrate from the gut, the mortar degrades. The wall becomes leaky.
And then things get through.
Precisely. It allows bacterial endotoxins, specifically lipopolysaccharides, or LPS, to act as invaders. They cross from the gut into the bloodstream, triggering a cytokine storm. This flood of inflammatory markers travels straight to the brain, activates the brain's immune cells, and causes the exact neuroinflammation we see in depression. We have the molecular blueprints mapped out.
Yes, we have them mapped out in mice, and I'm sorry, but I just don't buy that.
Really? You don't buy the Harvard data?
Let me tell you why. The historical reliance on Murrian models, lab mice, in this field has created an insurmountable translation gap. It leads to massive overpromising. You mentioned SCFA production in leaky gut. Do you know the anatomical difference between a lab mouse and a human being when it comes to digestion?
Well, they're different species, obviously, but
A mouse's small intestine to colon ratio is 2.5 to 1. In a human, it's 7-to-1.
The macroscopic anatomy differs, certainly, but the fundamental biochemical pathways.
But the fundamental anatomy dictates the biochemistry. Mice have a large, highly functional cecum that serves as their primary fermentation hub. Humans have a vestigial appendix, and we ferment entirely differently in our colon. The microbial niches simply aren't equivalent.
I mean, that's true, but we use specialized models to account for that.
Actually, it gets worse when we look at the models used to prove causality. Take human microbiota-associated rodents, the HMA models.
Right, the gold standard for causality.
Supposedly. This is where researchers take human fecal samples and transplant them into sterile, germ-free mice. But a comprehensive review in the journal Cell evaluated 38 of these studies. They found that human microbes undergo a massive ecological shift upon being placed into a mouse gut.
Well, there is some drop-off.
It's more than a drop-off. Only 4% of the bacterial genes share considerable identity between the two species. The community that ends up growing in the mouse doesn't even represent the human donor's pathology anymore.
And beyond the physiology, we are absurdly anthropomorphizing animal behavior.
In what sense? In the sense that mice do not experience human despair? Measuring how quickly a mouse gives up swimming in a beaker of water, which is the standard test for depression in these models, does not equate to the complex cognitive, emotional, and social framework of major depressive disorder in a human being. We're measuring a mouse's survival instinct and calling it human psychiatric disease.
I'm not convinced by that line of reasoning because it dismisses the fundamental utility of biological modeling entirely.
Oh, come on. A beaker of water isn't clinical depression.
Yes, a mouse is not a tiny human. We all know that. But think of a germ-free mouse model, like a wind tunnel in aerospace engineering.
A wind tunnel?
A wind tunnel is not the open sky. It is an isolated, highly controlled, fundamentally artificial and imperfect environment. But it is strictly necessary to isolate variables and prove the underlying physical principles of aerodynamics.
Okay, I see the analogy.
In this context, the germ-free mouse is our biological wind tunnel. It proves molecular capability. It proves that the Morganella DEA pathway, or the domino-like spread of misfolded proteins, or the LPS-induced microglial activation is biologically possible and causally potent. We use the mouse to isolate the variable and prove the mechanism exists, not to perfectly recreate the human emotional experience of despair.
That's a compelling argument. But have you considered that a wind tunnel tests physical laws of fluid dynamics that are universal on Earth? A host microbiome interaction, on the other hand, is entirely specific to the evolutionary biology and immunology of the host.
Well, sure.
A lab mouse kept in an ultra-sterile bubble simply lacks the lifetime of antigen exposure, the viruses, and the environmental stress that shapes actual human neuroinflammation. But fine, let's step out of the biological wind tunnel.
Gladly.
If we agree animal models have these translational limitations, we have to rely on human data to break the tie between causation and correlation.
I agree entirely. And the ultimate human tiebreaker here is Mendelian randomization.
Right, MR.
Because we obviously can't run controlled, germ-free experiments on human subjects. We can't raise humans in sterile bubbles. We use Mendelian randomization, or MR, to infer causality from observational data. It's essentially nature's own randomized clinical trial.
Because of genetic sorting.
Exactly. At birth, humans are randomly allocated host genetic variants. These are tiny shifts in our DNA that dictate a higher or lower baseline abundance of specific gut microbes throughout our lives.
Right, and the theoretical benefit is that they bypass environmental confounding.
Precisely. Because these genetic instruments are set the moment you are born, they are completely unaffected by whether someone later eats a depressed diet or starts taking Parkinson's medication at age 60.
Okay, so what does the MR data show?
The data is striking. It shows that a genetically driven higher abundance of a specific bacteria family, Coriobateriacea, causally increases the risk of Parkinson's disease by 1% for every unit increase. We also see that another bacteria, Desulfovibrio, acts as a causal risk factor by producing neurotoxic hydrogen sulfide. In psychiatry, genetic liability to depression causally reduces Bifidobacteriacea, indicating a self-perpetuating bidirectional loop. The point is, the human genetics prove the causal arrow points from the gut to the brain.
MR provides hints, certainly, but to call them unequivocal proof of causality is massively overstating the robustness of the data.
How so? The genes don't change.
Let's be transparent about what MR actually is. The human genetic instruments we use as proxies for specific microbes are notoriously weak. The variance in your microbiome explained by your host genetics is incredibly small compared to what you eat or where you live.
But the statistical signal remains valid.
But more importantly, Mendelian randomization in this field is completely plagued by pleiotropy.
You'll have to define that for the audience.
Sure, pleiotropy is when one genetic variant influences two seemingly unrelated traits independently.
Okay.
So the genetic variant that supposedly increases a certain microbe might also independently alter the brain's structural connectivity or tweak the immune response through a completely different biological pathway.
Right. Confounding the causal link.
Exactly. So you think the microbe is causing the brain disease, but really the gene is just causing both at the same time. And if you want a perfect example of how messy this gets, look at the coprococcus paradox.
Ah, the bidirectional findings.
Exactly. Mendelian randomization studies have shown bidirectional causal relationships with the coprococcus genus in Parkinson's disease. In some contexts, it appears as an early causal driver of inflammation.
Which supports my point.
But its abundance is also heavily altered by the disease process itself later on. It acts as a driver on Tuesday and a correlative marker on Friday. That level of complexity completely blurs the neat, unidirectional causal line you're trying to draw.
Well, it blurs the line, but it certainly doesn't erase it. Complexity is a feature of advanced biology, not a refutation of causality. But look, if you feel genetic hints aren't enough, we have to look at the ultimate clinical test of the driver hypothesis, interventional efficacy.
You mean FMT trials?
Yes, fecal microbiota transplants. It's a simple premise. If the gut drives the disease, then fixing the gut should heal the brain. And we are seeing that exact phenomenon in human trials.
The data there is highly contradictory, to say the least.
Let's look at the clinical successes first, because they are profound. Consider the 2026 China Phase 2 trial. Researchers took 72 drug-naive Parkinson's patients.
Which is a key detail.
Drug-naive is vital here, yes? It means these patients have never taken dopaminergic medications, so their guts weren't confounded by years of pharmaceutical side effects. They administered repeated donor FMTs, and the results were undeniable.
In terms of motor scores?
Yes, patients saw significant improvements in their UPDRS-3 scores, which is the standardized scale neurologists use to measure physical tremors and muscular stiffness. Even more impressively, physical biopsies of their colons showed a marked decrease in the clumping of that toxic alpha-synuclein protein.
That is a notable finding, yes.
And we're also seeing this in psychiatry. The Wong 2026 randomized controlled trials showed that treating depression with a combination of FMT and standard medication significantly outperformed medication alone. We are resetting the ecological foundation and measurably reversing the pathology in the brain.
Okay, but I have to counter that with the 2024 Finnish randomized trial for Parkinson's, which paints a vastly different, much more sobering picture of clinical reality.
The Finnish trial had issues.
It was a rigorously controlled double-blind study of 45 patients, and those who received the placebo transplant actually yielded better cognitive scores on the MoCA assessment, the standard test for memory and cognitive function, than those who received the actual FMT.
The placebo?
Let me repeat that for the listener. The placebo outperformed the targeted ecological transplant. Furthermore, the FMT group experienced a 53% rate of gastrointestinal adverse events, compared to just 7% in the placebo group. It made them sicker.
That Finnish trial had significant methodological limitations regarding the timing of the intervention. I mean, they were treating established late-stage disease.
The true limitation is the conceptual framework we are pushing onto the public. By framing the microbiome as this miraculous causal lever, we are wading into the deep sociological danger of cruel optimism.
Cruel optimism?
Sociologist Lauren Berlant coined this term,
And a brilliant 2025 paper in Nature, Humanities, and Social Sciences Communications applied it perfectly to the promissory language of modern microbiome science. For anyone listening who takes a generic probiotic pill every morning thinking it's curing their brain fog, this data is a harsh reality check.
I don't think it's a harsh reality check. I think it's just early science.
We are telling desperate patients that their neurodegenerative decline or their profound clinical depression is just a matter of, healing their gut.
But it gives them agency over their health. It gives them something actionable.
It gives them false hope. It drives them to dangerous, unregulated DIY fecal transplants. Or worse, it burdens them with unwarranted, crushing guilt when their incredibly strict dietary interventions inevitably fail to cure a complex, multisystemic neurological disease.
Well, no one is saying a diet cures advanced Parkinson's.
But patients internalize it. Oh, my Parkinson's progressed because I didn't eat enough fermented cabbage. When we overstate the gut as a causal driver, we elevate expectations to the level of a medical revolution without the standardized biomarkers to back it up. And that causes real psychological harm.
That's an interesting point, though I would frame it differently. The danger of cruel optimism is incredibly real, and I agree the scientific community must communicate responsibly. But that danger stems from treating FMT like a monolithic pharmaceutical pill, like an aspirin.
Which it's not.
Exactly. It's a massive category error. An FMT is not a targeted chemical drug. It is a full ecological transplant. The failure of the finished trial doesn't disprove causality at all. It simply highlights the profound difficulty of engrafting a healthy, vibrant, new ecosystem into a diseased, hostile gut environment late in a condition's progression.
But that just proves...
Let me finish. The China trial succeeded precisely because they used drug-naive patients in the early stages of the disease, before the internal ecosystem was entirely collapsed by altered motility and years of harsh medication.
But that brings us right back to my core argument about reverse causation.
How so?
If the gut ecosystem is so hostile and collapsed because of the established disease state, then the dyssymbiosis we are trying to treat is fundamentally a marker of that disease. Even if there is some minor feedback loop occurring, the clinical effect sizes we see in humans are incredibly modest.
Modest effect sizes still prove a link.
We're talking about standardized mean differences of 0.3 to 0.5. The placebo effects in these psychiatric and neurological trials are massive. Dysbiosis is primarily a reflection of disease-driven lifestyle, diet, and motility changes. It's a fascinating biological echo of what the brain is going through, but it is not the original voice.
And I maintain that the original voice is indeed coming from the gut, long before the brain hears the echo. I mean, the precise molecular mechanisms we've discussed, the invasion of LPS through a degraded intestinal castle wall, the domino-like spread of misfolded proteins up the vagus nerve, the DEA-driven immune stimulation from Morganella, these clearly demonstrate that the microbiome acts as a causative spark.
It's just a piece of the puzzle.
It is the catalyst that initiates the neuroinflammatory cascade, even if the brain eventually drives top-down changes much later in the disease cycle.
Well, despite our disagreement on what is driving the vehicle, we can at least agree on what definitely does not work.
Oh, absolutely. The commercial one-size-fits-all probiotic supplement market.
It is essentially a myth built on exactly the kind of promissory language I warned about. Throwing a generic lactobacillus capsule at a complex neurological disorder fails because those transient bacteria simply cannot establish a permanent ecological niche in a mature, resident gut ecosystem.
Right, it's like throwing a single seed into a dense forest and expecting it to take over.
Completely agreed. And while we are correcting the record, we also have to correct the lingering, outdated scientific myths that still dominate public perception in 2026.
Oh, please do.
The bacteria-to-human cell ratio is not 10 to 1, as is so often cited in pop science articles. It's roughly 1 to 1, about 38 trillion bacteria to 30 trillion human cells.
Yeah, and the microbiome does not weigh 1 to 2 kilograms. The total mass is really only about 0.2 kilograms. Precision in our language and our data matters immensely if we want to move this field forward into standardized clinical practice.
It absolutely does. And moving forward, I believe our focus must shift toward what's being called the lifestyle microbiota health nexus.
Right, looking at environmental mediators.
Yes, the microbiome is an incredibly sensitive mediator of environmental toxins.
A 2025 study from the University of Cambridge identified 168 common chemicals in food and household items, things like flame retardants and pesticides, that act as literal poison to beneficial gut bacteria.
It's terrifying to look at.
Our focus should be on how everyday chemicals degrade this delicate ecosystem, rather than viewing the gut as a magical biological switch we can simply flip to cure the brain.
That is a crucial point of convergence for both of us. The truth of the microbiota gut-brain axis likely lies in a highly complex, multisystemic feedback loop. The gut microbiome undeniably provides a window into a body that is already ill. But through approaches like precision nutrition, specifically utilizing interventions like the Mediterranean diet to synergize fiber and polyphenols for targeted short-chain fatty acid production, it potentially holds the lever to help the body heal.
As long as we recognize the vast complexity that's left to explore, and temper our clinical promises with rigorous, standardized science.
Exactly. At the start of this hour, we asked if the gut was the arsonist that started the fire or merely the smoke alarm reacting to it.
Or maybe a bit of both.
Right. Perhaps the most accurate view is that the microbiome is the oxygen in the room capable of sustaining vibrant health or fanning the flames of neurodegeneration once the initial spark is lit. We'll leave it to you, the listener, to examine the evidence and form your own conclusion.